Alright, welcome back. And in our last video, we spoke about simple sugars, monosaccharides and disaccharides. In this one, we're going to look at how those simple sugars, the glucose, fructose, or galactose monomers, primarily glucose in this case, come together to form large polymers and what that means in terms of their structure, where we find them, and their uses. So polysaccharides, as the name suggests, are carbohydrates with large numbers of saccharides. So ones with less than 5 monosaccharides are called simple carbohydrates, i.e. 5 sugars or under put together. More than 5 put together are called complex carbohydrates or polysaccharides. Unlike mono and disaccharides, polysaccharides are non-crystalline. Okay, so our monosaccharides and disaccharides tend to form crystalline structures, as we know from the way that we see sugar when we use it. Okay, remember that's sucrose, and that tends to be a crystalline structure. Okay, they're soluble in water, as we talked about, and they have a sweet flavor. Polysaccharides, however, are non-crystalline, so they don't tend to form crystalline structures. They are generally insoluble in water. The large polymers are insoluble in water, remembering that we've lost hydroxyls every time we have a condensation reaction. Okay, so there are less hydroxyls across the entire molecule due to the condensation reactions than there are in the monosaccharides. So this in itself gives a lack of solubility, as well as the secondary structures that we tend to see. Okay, the other thing that's interesting about these is that polysaccharides do not taste sweet. They are largely tasteless. The 3 most common and most biologically important ones, particularly for us in our study of VCE chemistry, are starch, cellulose, and glycogen. These all differ in the way that the monosaccharides are put together, but they are all polymers of glucose. Okay, so we can see here starch tends to be more straight chain. Cellulose, we have alternating structures. This is our CH2OH, which you can see alternates. And then glycogen, we have a more branched and open structure. So each of the complex carbohydrates differs in these basic bonding structures. For each of the polysaccharides, the level of branching in the polymer chain can be used to explain the overall properties, the function, and the rate at which they are hydrolyzed within our body. Okay, so cellulose tends to be straight, linear, and highly packing. Starch can be branched, as we see, as well as less branched. We will look at 2 different types of starch, amylose and amylopectin. And then the more highly branched is going to be glycogen. Short branches, many of them. So when we talk about starch, starch is the polysaccharide that we find in plants. Okay, they're able to produce alpha glucose from sunlight via photosynthesis, and then they polymerize this alpha glucose to form starch. So starch is the energy store of plants. Okay, so it's the polymer that plants lay down to store energy. It's found in the fleshy parts of fruit and vegetables, particularly ones that we consider high starchy vegetables, such as potatoes and corn, things like that. The simplest form of starch is known as amylose, and this is a linear or straight chain polymer. Okay, we can see that all the glucose molecules here are orientated in the same way. They're oriented in the same way, and we have a 1,4 glycosidic link for each glucose monomer forming into the polymer. So some condensation reactions occur between the hydroxyl groups at different positions. Okay, some branching of the starch can occur. When we see this, we get a slightly branched version of starch that we refer to as amylopectin. Okay, so amylopectin is a branched form of starch, whereas amylose is our straight chain. And the structures for these, again, are given in your data booklet. So amylose is a straight chain polymer, and what we tend to see is it forming these helical structures that are quite tightly packed. Okay, so we have tight packing. What this tends to mean is that it can be hard for enzymes to get into here. Okay, so this is broken down by the enzyme amylase in the mouth. Okay. Amylopectin is a branched polymer of starch. Okay, so we have a more random, less linear polymer that we see with amylopectin. Cellulose is the structural polymer of glucose that we find in plants. Cellulose is only found in plants, and there are very few organisms that can break down cellulose into its component glucose monomers for energy. It's the most common carbohydrate. Okay, it's the main structural component of the cell wall. So cellulose is formed by the polymerization of beta glucose, not alpha glucose, and is made up of parallel linear polymer chains. Because of this polymerization of beta, we have an alternating CH2OH on either side. This alternating structure allows it to form quite tightly packed linear chains that form parallel to each other. Okay, this allows for good alignment of the hydroxyl groups, strong hydrogen bonding, giving us a strong fibrous material. So cellulose is not used for energy, it is used as the structural polymer in plants. And as I said before, very few animals have the cellulase enzyme capable of breaking this down. Here you can see the way in which we get strong hydrogen bonding between the groups in the glucose monomers. Also remembering that it is beta glucose that forms these polymers, whereas starch is alpha glucose. Glycogen is the equivalent of glucose storage for animals. So starch was the energy storage for plants. Okay, and we said that we could have the straight chain amylose or the branched amylopectin. When we look at glycogen, you can see that there are a lot more multi-branched. It's a multi-branched polymer of glucose. This means it has even less structure. Okay, so it tends to be less rigid. So it is a multi-branch chain that is produced in animals to store excess glucose. It's stored in the liver and in muscle tissue until needed, and then it's consumed above when the glucose needs of the body are above what they can produce. If this continues, then it's converted. If the excess consumption of carbohydrates continues, then the glycogen is converted into fats and lipids later on. Okay, so fats and lipids are our long-term storage. Glycogen is sort of our short-term storage where it's going to be used up in our muscles. So this is what we're trying to build up when athletes are carb loading. They're building up that excess store of carbohydrates, and that's stored as glycogen. The glycogen is a polymer of alpha glucose, so similar to starch, made up of the same monomer, but it is more branched than amylopectin. This is the structure that you will see in your data booklet, okay, and you can see here that the branch tends to come off carbon-6, this CH2OH, turns into a CH2, and then we have a condensation reaction between the OH hydroxyl and another hydroxyl of a glucose monomer that will then polymerize itself, okay. So as you can expect, the structure of these polysaccharides alters their solubility. The long chains of amylose coil into spiral-like helices with the hydroxyl groups pointing inwards. If the hydroxyls are pointing inwards, this means that they are not free to be interacting with an aqueous environment. This makes them largely insoluble. In contrast, the branching in amylopectin restricts coiling, so this means that the hydroxyl groups and the will form hydrogen bonds with water, increasing solubility. Amylase and the enzymes that break down starches are aqueous bound enzymes. So the more open structure of amylopectin tends to lend it to being broken down more rapidly, whereas the helical structure of amylose can reduce the rate at which it is hydrolyzed in the body. Both forms of starch can be broken down, unlike cellulose, which is pretty much insoluble in water because of its tight packing, making it even harder for enzymes to get to. Ruminants such as cows have cellulase and multiple stomachs to allow for the digestion of these sugars from cellulose. Okay, let's have a quick look at a VCAR question and see the way that these tend to form. You can see here that they have given us the structure of a disaccharide and they've told us that it is maltose. You should remember that maltose is a disaccharide of glucose, and you can check your data booklet and confirm this from the structures that you are given. You are told to draw the structure of the monomer from which maltose is derived. So what you're going to do is break the glycosidic link and essentially straighten this one up and add a hydrogen onto here. So we're just going to copy it out. 1, 2, 3, down to our oxygen, over. So we make sure we've got a 6-membered ring. Then where we broke the glycosidic link, we're going to draw our OH, H, this one has above the ring, hydrogen below, the next one around is an OH with a hydrogen here, OH, hydrogen, H, CH2OH. And this is in fact glucose, alpha glucose. What is the name of the monomer drawn in part A? It is alpha glucose. Identify the type of reaction that occurs when these monomers combine to form maltose. So when they are combining, it is going to be the condensation reaction. It's not polymerization. I'm going to remove that because it's just a disaccharide. So it's a condensation reaction. If it was starch, I would write polymerization. Name the linkage joining the monomers in maltose. This is a glycosidic link, and you do have to spell this correctly. And if we have a look, there we have. If you have trouble spelling glycosidic, you can refer to it as an ether link. This is a valid way to answer this question. Okay, that's it from me on our polysaccharides. We will look at some more questions in class and the way that these link in with our lipids and our peptides, as well as questions on enzymes.